after the coffee break, we will have the last lecture. And we'd like to invite Dr. Alexandre Nepomuceno. And Dr. Alexandre is agronomist. He took your master from Federal University of Rio Grande do Sul. Took your doctorate degree at molecular biology and plant physiology from University of Arkansas. Postdoctorate at the Japan International Research Center for Agricultural Science. Has experience in plant physiology and plant molecular biology, working with gene expression, soybean physiology, biosafe GMO, and genetic engineering. Please, Dr. Alexandre.
Thank you very much for the presentation. I'd like to thank you, the organizers, specifically Dr. Luis Avila, that invited me to come here and talk a little bit about genome edition. We are living a revolution in genetics and now in genome edition. And Brazil and many countries in the Americas are having a very excellent opportunity to start to develop products where you use biotechnology, products that can help to solve problems in agriculture or aggregate value. Because before of that, we could not do that. In the transgenics area, in the GMOs area, unfortunately, today only a few companies can put products in the market. And my talk will be much more regarding this. I'm not going to be giving details about genome editing, to explain just an overview about how the technique works. And just a little bit about weed science. I told Avila that I'm not a weed science guy, but I think it's interesting for you to understand a little bit these tools and how the legislation is changing over the world, facilitating the use of this technology. Just an introduction. I don't know to explain this for you. In the last 40 years, the production, it's too loud? The production in Brazil in the last 40 years increased more than 600%. And we have just an increase in 160% in air. That means lots of technology. The transgenics was a very important tool, not the most important. We have no-tillage, soil management, insect control, all the other techniques. Transgenic was important too, mainly in the no-tillage strategies. Today, I forgot to mention, right, I think it's important for my talk, I am also a member of the Brazilian Biosafety Commission. I'm in the 8th mandate, so I'm going to finish my mandate next year. But I have been following all this discussion about GMO technology and genome edition in Brazil for the last 15 years. So it's important to say that. But in terms of GMO technology, today in Brazil we have more than 170 genetically modified organisms. Liberate, we have more, the most part is in plants, 60% plants, vaccines, microorganisms. Brazil was the first to liberate commercially an insect to control diseases, a genetically modified mosquito to control the Aedes aegypti gene, and Aedes aegypti mosquito, and it's working very well in the cities it was liberated. But because of this big discussion about GMO, in Brazil we have a kind of moratorium from 1998 to 2006. The Roundup Ready soybean from Monsanto was liberated commercially in 1994 in the US, 96 in the US, 97 in Argentina, and 98 in Brazil. In US, it was 40–50% adoption by the producers in the beginning. Today is almost 100. In Argentina, from day to night, became 100% adoption. Because in Argentina, they do not sign the TRIPS treaty. That's their treaty for international recognition of patents. That's why they start to sow Roundup soybeans. But the problem, and in Brazil, CTN Bio liberated in 1998, but then Greenpeace, IDEC, and IBAMA, a Brazilian agency for environmental protection, ingress in the justice and block the use of the technology. And because there was a conflict between the old biosafety law and the environmental law in Brazil. And then depending of the judges that look at the discussion, it liberate or block, liberate and block. In practice, during 7 years, we could not use the technology in Brazil. So we start to see the Maradona soybean coming from the south, varieties not adapted to our conditions, no investment in research, the seed producers start to lose money, so it caused lots of trouble in Brazil. But for me, as a scientist, maybe the biggest problem was that we do not invest in this technology during 7 years, basically. Okay, there was no support for the financial agencies to do research in GMOs, or just a little. So we are seeing that now. And then, because of this conflict between both laws, biosafety, the old biosafety, and the environmental law, in 2005 came the new Brazilian biosafety law, and then new rules were applied, and then we started to see you can see here from 2007 is when it started the commercial liberation of many GMO, mainly plants in our country, helping the agriculture in many cases, causing trouble in others. But it is a very interesting technology, and a country like Brazil must be able to use it. Okay. The first one, I don't need to explain. Everybody, I believe, in this room knows how Roundup Red works. Ammonium glyphosate, also plants with genes, all transgenics. You get the whole gene from from another species. In the Roundup Ready and the ammonium glyphosate resistant plants came genes from bacteria, the BAR gene, the PAT gene, the CPS, the EPS4, the EPSPS, the 5-enol-pyruvyl shikimate trisphosphate synthase gene. That's why we call EPSPS. And then came the Roundup Ready 2. Actually, there is a dispute right now in the Brazilian justice between Bayer now, right? Because bought Monsanto, and the AproSoja, the soybean associations producers in Brazil. They are discussing if there is no innovation in the Roundup Ready 2. Last year, the Mato Grosso State Justice just gave win to the AproSoja. Of course, the Bayer is disputing that. But just for you to have an idea, the Mato Grosso Justice told Monsanto to pay 2.4 billion reais for the producers in Mato Grosso. They are disputing that, right? And if you have time, I can give you details about the difference of those two GMOs. Now you have the Intacta Extend with dicamba, and the soybean, both for soybean. Actually, I'm sorry, dicamba is just for soybean. I didn't see for other crops. And the enlist you have for soybean and for cotton. Last week, I was in Brasilia at the Brazilian Biosafety Commission. We just liberate the last enlist cotton to be planted in Brazil. But there is a big discussion, probably you are aware of that, I'm sure you are aware of that because you are with science guys, about the discussions regarding deriva, volatilization and drifting, actually, in those crops. They are not planted in Brazil yet, but probably when they start to be planted, maybe they're going to cause some trouble. That's my personal view. And now in Brazil, in terms of GM, genetically modified plants, where they got genes from one species and put in another, you have all this, I was in the planet, I forgot to translate, but we have all these genes in plants that are authorized to be used commercially in Brazil with the resistance for those genes. Then, after 2005, with the new Brazilian Biosafety Law, we start to plant genetically modified plants in Brazil, and today we're the second country in terms of production of GM plants. But it was created this paranoia over the transgenics. Although we use the transgenics, started to use transgenics late in the 70s for insulin, growth hormone, some detergents that we use in our kitchens, vaccines, many products are based in genetic engineering. But the paranoia actually started with when we started to plant Roundup Ready. There is many reasons for that. Maybe biosafety was not the most important, but I will spend the whole day discussing that. Maybe in the questions we can talk a little bit about that. But was created this paranoia. And in practice, actually this is a poster. You can see my picture here. In 2007, they spread these pictures all over Brazil saying that those doctors are in favor of transgenics. They are members of the Brazilian Biosafety Commission. If you look at the face of the doctor, he's a crazy guy. But it was a really complicated time. Here we are discussing the liberation of the Liberty Link corn. The Greenpeace invaded our meeting. It was very difficult times. I put this because actually the villain, I believe I put between commas, but the villain, I believe, was Roundup Ready. For many reasons that I don't have time here to discuss. Of course, you change. You're using 1, 2, 3, 4, 5 different kinds of herbicide molecules, and from day to night you start to use just one. Many other herbicide molecules stop to be sold. So, but I can consider that the Roundup Ready was considered the villain of all this history in terms of GMO use in the world, not only in Brazil, in the world. And this, you probably cannot see this picture, but this is a paper from McDonald 2011, and actually Prado et al. 2014. Those are Monsanto scientists, now Bayer scientists. They got a study talking with all the other companies that were developed, the big companies that were developing GMOs, and they look at the cost to put a GMO in the market. From identifying a gene, making the proof of concept in a model plant, making the proof of concept in the crop that's your target, to start the tests in the field and put in the market, they estimate that costs $136 million. I don't believe it's that high, but it's high. Because of the paranoia over the transgenic, make every country in the world to make its own rule to regulate that. And that made very, very expensive for other groups to use the technology. Most of the cost of putting a GMO in the market is deregulation, mainly because all these, the reporter at Embrapa Soybean tells me, don't use the word paranoia, but it was a paranoia that was created over this technology. Although, like I said, people are using that in medicine and in the industry for years before the Roundup Ready. Because it's a GMO, takes too many rules, also takes longer to put in the market. For example, in Brazil, our legislation, if I developed at Embrapa a drought-tolerant soybean, that's what I'm working with, I need to test in the field for many years before to apply for a commercial use. So to test in the field, I need to ask authorization. If I have in the field, I have fiscalization, I have officials from the Ministry of Agriculture, from the Ministry of Environment coming and checking, and if I don't do something wrong, they apply a penalty to me. In practice, what happens? If you look at the CTNBU's agenda in the last few years, you don't see any Brazilian university or institution. You see Embrapa, we have us with soybean, we have Embrapa Agroenergy with sugarcane, and that's it. Because it starts causing, inhibits people to do this kind of work. So we do not develop technology for our situations. And then comes the CRISPR technology, the genome edition technology. Actually, genome edition is not that new. I'm going to show one example for you from this kind of technology that is one of the first herbicide-resistant plants from the Cybus company. But since the 80s, the early 90s, we are using genome edition, but with techniques much more complicated that I do not have time to explain here. I'm just going to give my own example that when I was working, I was coordinating the LabEx program of Embrapa. LabEx is Embrapa virtual laboratory overseas. We make partnerships with other institutions. And in my case, we did a partnership with USDA in Albany, California, at the Plant Gene Expression Center. So I was coordinating this action during 2 years. And there we are working with talents. Talents is a kind of genome edition technique. Very interesting, but it's very complicated. It's protein linking to DNA. Again, it's not my objective here to go to specifics of the technique, but I spent 4 months building my talent strategies, a piece of protein that connects to the gene I was trying to turn off. The second talent for one strain and to the other strain, the fork enzyme that cuts the DNA. Took me 4 months to build this structure, the talents, to turn off one gene, to cut one gene. And then we are there talking with the colleagues, and then we start to hear about CRISPRs that the Jennifer Donna was working at the Berkeley University. I didn't meet her, but I met many of her colleagues. And then we said, well, let's try this CRISPR thing. Took us 4 months, and we spent around $10,000, something around $10,000 to make this construct for talents, to turn off one gene. To turn off the same gene using CRISPRs took me 8 days between designing the CRISPRs and receiving them in the lab, and cost around $600 to do the same thing. That's why everybody is hearing about CRISPRs, and CRISPRs, because it's faster, it's easier, in some ways it's more precise. Of course, it's not trivial. It's not that easy to make edition in plants, but compared with the other techniques, it's much more easier. And in the case of CRISPRs or any other genome edition technique, let me go to the law first. We are lucky, at least in that point, we are lucky in the new Brazilian Biosafety Law 2005, our Congress gave us the mandate, I mean the CTN Bio, the Brazilian Biosafety Commission, the mandate to follow up innovation and propose new legislation to promote growing and development in Brazil and preserving biosafety. Using that, and also this, the Brazilian biosafety law exempts mutagenesis. And there is not a detail in the law explaining what's mutagenesis. Then, the Brazilian biosafety law use its concept in terms of what's to be considered mutagenesis. And when you use a technique like CRISPR to do a specific mutation, many times mimicking what nature does, in some cases, we consider that product that was generated by this, the use of this technique a non-GMO. In a case-by-case under consultation. Case-by-case under consultation. Why am I freezing that? Because in Argentina, it's a little bit different. In the US, it's different. Canada is a little bit. I'm going to try to explain a little bit of each one of the countries, but all are aligned. All consider, in some cases, the use of these techniques as a non-GMs. And then, it's in Portuguese, but in January 2018, Brazil published its regulation regarding genome edition, that in a company, an institution, a university that develops some product that uses this technique, submit a consultation to CTN Bio, and CTN Bio is going to say it's GM, it's not GM. I'm going to show some examples to you. Basically, what you ask, which technique you use in the seedlings. You're going to have exogenous DNA there, so use gene drive technology. We ask many questions. I suggest you to take a look at the Resolução Normativa número 16, 16 Normative Resolution at the CTN. It is very interesting. Why? Because it's not only related to genome edition. The initial focus was genome edition. But for example, the use of RNA, topical use of RNA, not as a GM plant, but for example, a purified GM RNA that I apply as a spray in a plant or in another organism, depending, again, case by case, can be considered a non-GM. Because in some cases to produce, for example, VIX it will be considered a GM here in Brazil. But you can use a bacteria to produce RNA and then purify the RNA and have the pure RNA there. Then you apply as a, let's say, insecticide or herbicide. But the resolution 16 goes in this direction, so I suggest you to take a look. Well, Argentina, and you're going to see how important it was to Argentina, was the first country. They started to discuss this in 2015, and they published in 2016 the first resolution regarding genome edition. Then Chile. Chile is using this a lot in fruits. Nobody wants to have a transgenic fruit, but you can use this technology, for example, to make fruits without seeds, using genome edition to delete a gene that is involved in seed production. Chile is also case by case, you consider a non-GM. Colombia, also. Paraguay, just 1 month ago. Uruguay probably is going to come out soon. Japan, Canada, and Australia are in the same way. Mainly in the SDN1 strategies, they are considered not GM. I'm going to explain what's SDN1. This is a picture. I have a meeting, and I was in Belgium, in Brussels last week, but actually I took this 2 years ago, but the USDA confirmed now in the United States, the companies, it's voluntary, the consultation is voluntary. If some company is doing genome edition in plants, not in animals, okay? Some company is doing genome edition in plants and causes SD1 mutation, that's the simple one, like nature does, and they consider a non-GM, they do not need to go to the government to ask the government, is GM or not GM? It's their responsibility. It's different. Canada is like Brazil and Argentina, the companies supposed to come to us and ask. The difference in Brazil, because of our, how to translate, lei da transparência, I don't know. Because of our laws, we must inform the public that the company X came to us and applied for this, ask CTNBio to ask if it's a GM or not GM. But because of intellectual property law, we do not need to tell to the public where was the mutation. If you do not know where you did the mutation, mainly in the SDN1 and SDN2 cases, you cannot detect. If you don't know where you did that, you cannot detect. It's an important thing because, anyway, later I will go for that. This is an overview picture of what's going on in the world. So like I said, Brazil, Argentina, Colombia, Paraguay, and Chile. Canada, Israel, United States, Philippines, Japan, Australia, and Uruguay is almost coming out the proposal. In green are the countries that is like Brazil. We're gonna analyze case by case, and depending of the case, we're gonna say that's not GM. It could be caused by a natural mutation. I could be obtaining that by crossing those two individuals, or I could cause that by irradiating those plants. But in this case, you did with a high precision by knowing the genetics of that species, and then use CRISPRs or whatever talents to change a little bit that gene. Again, it's going to be case by case, because some case we might be concerned of some pathway that was altered and may cause some trouble. But there is many examples I'm going to show you in that case that already exist in nature. Okay? Well, Europe, yeah, is complicated. There was a decision of the Court of Law last year, and they decide all mutagens are transgenics, except those ones did in the 40s and 50s and 60s with irradiation. Those are no GMOs. There is more than 2,000 varieties. Some are already out of the market, but varieties that were generated using radiation to cause point mutations. Think about, use radiation to cause point mutations all over the genome in plants, and then to sow those seeds, for example, in a dry soil and try to see a plant that is gonna grow. So, well, maybe I turn on or off a gene with a point mutation, an inversion, whatever, indel, that caused that trait that made that plant drought tolerant. No country in the world regulated this kind of technology, okay? Now we are doing the same kind of mutations, but with much more precision, knowing the genome we are changing, okay? So why consider GM? That's the big discussion. I've been participating with representatives of the European Union in the OECD meetings last April in Paris, 2 weeks ago in Belgium. It's very interesting when we talk with representatives of UK, France, Germany, everybody. We are using the technology, it's important the technology, but the decision of the just in Europe consider everything as a GM. They are trying to change, there is a movement to try to change that. I hope you can change that. And the other country that I have been participating in many meetings, 6 actually, is China. China is another big question because the Biosafety Commission of China have more than 70 representatives. It's a mix about between among scientists and the politicians. So depending who you talk with, goes in one direction or another. However, China is the country that's more publishing and making patents, applying for patents over genome edition technologies. Me avisa quando faltar. That's when was that decision last August? Bayer and BASF published that. I mean, that they are getting out of Europe to do genome editing. It's interesting because some companies are starting to apply to do research here in Argentina and in Brazil. I'm going to show you some data. If you want to read a little bit more about the situation, it's a paper. I am one of the authors with Dr. Erickson. And then my slides will be available so you can find out the paper. So we give an overview about what's going on in terms of legislation, but we are giving many examples of commercial liberations. Né? I don't like to use commercial liberation. I mean, I like to say that that country considers that variety as a conventional variety. That's the language we are using at the CETEN Bayer here in Brazil, Argentina, US. So you can, if you look at the paper, you can see examples. Well, I don't know, probably many of you here is not from the molecular biology field. I don't want to give you a class in molecular biology, but a very quick overview is like CRISPRs. Is like you have a drone, and in the GPS information of the drone, you put the sequence of the gene you want to cut. That's what CRISPR does. It cuts DNA, the whole system, the CRISPR-Cas9 system, right? So in the GPS information, you put the sequence that's very specific, 20, 25 base pairs of the gene you want to cut. And in the other part of the drone, you put a scissor. The scissor, tesoura, the scissor is going to be the Cas9 protein. It's a nuclease. You have new ones now. You have CMS1. Embrapa just did one agreement with, we closed an agreement with the Benson Hill company. They have many other nucleases. CMS1 is one of those. CPF1 is another enzyme. Mainly for plants, those enzymes are more interesting because the Cas9 is represented here by it will be the nuclease, the Cas9. In here, the CRISPRs, the gRNA, the guide RNA, where you put the information in the GPS, and this whole system go to the nucleus and start to screen the chromosomes. When it finds a sequence homologous to that sequence of the gene that you want to cut, it goes there, the Cas9 goes there and cut. When the Cas9 cuts DNA, many things can happen. In the SDN1, site-direct mutagenesis, nucleases, site-direct nucleases, you cut DNA and the evolutive mechanisms in the cell try to fix it. Try to put the two strands together. By doing that, sometimes it erases some bases, or some bases are added. And depending how it happens, it changes the reading frame. Everybody knows that the genes are read by codons. The proteins, the amino acids are 3 bases, codons, right? They're read 3 by 3. If you take off one of those bases, you're going to have a change in the sequence of the codons. And then you're going to have all other amino acids, probably going to have a stop codon, and then that protein stops working. Okay? SDN1 systems are like that. You cut and you leave the cell to fix it, and sometimes you're going to have a mutation, a deletion, whatever, that's going to turn off that gene and going to change that pathway, and that pathway going to give some interesting trait, for example, herbicide resistance. The SDN2 systems is when you cut the DNA, and at the same time that you open that strain, you put there a template, a DNA template, with the mutation or mutations you want to introduce in that gene. You use a sequence of DNA with homologous sequence, then you cause homologous recombination by chance here, and in some cases you're going to have this homologous recombination introducing the mutations that you introduced here in your DNA. That's the SDN2, the most discussed systems today. Usually, SDN3 systems, you have the whole gene, promoter, coding, terminator. Usually, all countries are considering that as GM. But still a big discussion because in some countries, depending what about if I may use the DNA from the same species? For example, glycine-max and glycine-max should be considered. But the big discussion today is about these two. Brazil, Argentina, United States, and Canada, Chile also, are considering when you have DNA from the same species, like I said, so glycine-max and glycine-max, Saccharomyces cerevisiae and Saccharomyces cerevisiae, using pieces of DNA, you're going to consider at least Brazil and Argentina, in the case we have. I know that US have a case like that too, are considered as non-GM. One of the discussions is about the size of this fragment. Let me see in my next slide. Yes. I was in the OECD meeting last April in Paris, and every country was presenting. And then the Australia guy said, well, SDN1 in Australia is going to be non-GM. SDN2 depends of the size of the fragment. Big size, it'll be GM. Small size, going to be non-GM. And then I ask, well, what's a big size and what's a small size? For example, soybean genome has 1 gigabases, right? 1,000 base pairs for me is small. So he could not answer. And I believe this discussion disappeared now. It's more now the discussion is over if it's the template in the SD2 systems, the template is from the same species or from exotic species, exotic DNA. Then it's going to be considered GM, and in case you are using the same species, non-GMA. Again, case by case in most of these countries, exception US. This paper came out because we are having a big discussion at Seteno Bio, showing that many of these inversions, deletions, insertions of big pieces of DNA usually occurs in nature, inside of the same species. I suggest you to look at this paper. So we are doing things that nature does, but now with a strategy, with more precision using these techniques. And in some cases, we want to say those plants, those microorganisms are not GM. In Brazil, you are doing lots of work. Embrapa is investing a lot on that. For herbicide resistance, I'm going to show an example we are doing in our lab. In our lab, we are trying SDN2 system to make soybean plants resistant to imidazolinones, imazapyr, right? Yeah. You know that many of the mechanisms of resistance, not all in weeds, come from point mutations, mainly enzymes involved in the mechanisms where the herbicide turns off the enzyme. So if you have mutations, like you have in the CP4, EPS-PS genes from the glyphosate, you have mutations that change one amino acid, changes a little bit the structure, you might have resistance if the herbicide cannot connect there. Of course, it's not that simple, but in many cases like that, like was presented in the other talks here, we have many mutations known in nature, and now it's becoming easier and easier to identify those mutations because sequencing DNA is getting cheaper, faster and faster. Before we need to have a reference genome, and now we have these new systems from nanopore, PacBio sequencers that you can sequence long strains of DNA with more than 20 megabases, 8 megabases in 10 minutes. Whoa. So let's accelerate. So as you know, then those mutations can cause resistance to those herbicides. One of the first ones in the 90s was this Cybus example where they use this. It's a patent technology from the company Cybus. I don't know, it's works like a PCR. Probably some of you know how a PCR technique works. But they use a kind of primer to change the sequence and introduce a mutation. By doing that, they develop canola. Canola in English is? I don't remember the name. Say hi, louder. Rapeseed. So rapeseed resistant to imidazolinones, okay, using this technique. It is being planted in the US. It has been planted in Canada. If you go to their homepage, the Cybus company is saying there, non-GM. And then Cybus company went to Germany. The German government said, no, that's not GM. It was a mutation that happens in nature, you just use. And then some non-governmental institutions complain about that. It's under dispute now in Germany. I don't know what's going on right now. And then you have this decision from the Court of Law. This is in that paper I showed you, some of the genome-edited crops, oilseed rape, soybean, Comelina sativa, with herbicide, different kinds of herbicides using different strategies. And there is a soybean drought tolerant that also was authorized. It's not planted yet, but was authorized as a non-GM in the United States. In our case, as you know, imidazolinones, Embrapa with BASF during 20 years tried to develop, and we developed a soybean variety resistant to imidazolinones, but because it was a GM plant, it was a gene from Arabidopsis thaliana in soybean with the mutations. With that but because it took so much time, it was much longer than we expected, it lost its time. This technology will never go to the field. We decided that there is better technology, it's not worth to go ahead. So anyway, as a proof of concept, because SDN2 systems is not easy to do, SDN1 is much easier, SDN2 is much more hard. There is some groups getting good results. Dr. Kaxia Gao from China, she's a partner of us in a project for Brazil, China, in soybean. She's doing that in wheat, and we are trying to use her technology to do that in soybean. We are trying to do what? We are using 2 CRISPR systems to introduce a template of DNA. The whole sequence here is the same of the flanking regions of the sequence where we cut with CRISPRs. Then we cut with the CRISPRs, and then we introduce a template with 2 mutations that give resistance in soybean for imidazolinones. We are using to prove the concept because we want to use the same technique, and with the herbicide resistant is easy to see in the tissue culture, but you want to use the same concept to change promoter regions in many genes in soybean that you know that we change the expression of that gene, increase the expression, improve drought tolerance. And we are trying to do that. In this case, in Brazil, Argentina, probably US, Canada, you would be considered a non-GM. Of course, if in the final product you have only this mutation, those 2 mutations introduced. I'm gonna speed up. I mean, this is one company in Argentina that started to do this. They are a startup. They were 2 weeks ago. I was in the Mercosur in Argentina. They presented their results with herbicide resistant using genome edition, and they're planning to put this in the market pretty soon. In Brazil, we don't have any herbicide resistant plant that uses genome edition submitted yet, but you have this yeast, but it's a good example so you can understand why it's important. a legislation that keeps the biosafety, preserves the biosafety, but allows other groups, smaller, medium groups, to also participate. This yeast, it was developed by a Belgian-Brazilian startup. The president is a Brazilian guy. They have a yeast used to produce alcohol from sugarcane, and then they look at the yeasts, Saccharomyces cerevisiae. They look at the Saccharomyces cerevisiae used to produce sake, you know, the Japanese drink, and they identified 4 mutations in 4 different genes that improve the production of alcohol. They use CRISPRs to transfer those mutations in a SDN2 system to introduce those mutations in Saccharomyces cerevisiae. CTNBio analyzed that, and the final report says, you could be obtaining that by crossing those 2 yeasts. You take longer, much less precision, but you could reach the same result. You could obtain that by applying radiation. Probably it would take forever, but you could. In theory, you could. So we consider this yeast a conventional yeast. If they apply to the Brazilian biosafety 2 years ago, when you do not have this new resolution, it would cost them $400,000 to do all these deregulation documents and studies to do that, to get authorization as a GM for commercial use. So it makes a big difference. Now they had 0 cost for this deregulation. Of course, they have other costs. This is an important case because Corteva, everybody knows Corteva is down the pole in Pioneer, became Corteva. They are using this waxy corn to test legislation all over the world. They apply first in Argentina, US, Canada. Argentina, US, Canada already said. Let me very quickly, I know that my time is, usually starch from corn have about 25% amylopectin, 25% amylose. There is many mutations known in the nature. Early, last century, people identified with these waxy corns that they have mutations in this amylose, alpha amylose gene that turns off the gene. And then you have 100% amylopectin starch in those corns. There is many kinds of mutations. Why it's important, this kind of starch? I don't work in this area, but they say that it's good to make paper, glue, to use in the industry in general. What Corteva did, they use CRISPRs, and they delete about, I don't think I'm supposed to say okay, what they delete, but they delete a piece of the gene, and then the gene was turned off. The same kind of mutation that is already known, the same kind of result is already known that happens in nature. What they did this time, they use a high yield producing corn to do that. They could do that by getting those corns that are already in the nature and by breeding, crossing. It would take many years, not with the same precision, but maybe they could get to the same result. What they did, they use a high producing corn, and they use that CRISPRs to do this deletion. CTNBio analyzed that and said, the same thing for the yeast I presented before. It's a conventional, could be obtained by breeding, it's not GM. Can you imagine if Brazil say, no, use a genetic engineering technique. It's GM. Then you're going to have Argentina planting, sowing, side by side with us, but the Brazilian producer would have to segregate, to rotulate, and then the cost. So, thanks God, my colleagues that analyzed that. It was, I think it was, the yeast was unanimous, the vote in the CTNBio is very rare. And for this, I think was 21-2 against. something like that. It's interesting. Argentina authorized as a conventional fish, this tilapia. They change a myostatin gene. Myostatin is a protein, is a kind of hormone everybody have. When you have low levels of this protein in your blood, you have a more formation of muscles, right? It is a known mutation in nature. This is not modified. This is the Belgian blue strain race. It's a mutation that already exists in nature. By understanding the genetics of this mutation, of these animals, they identified that the mutation of this myostatin, the turning off of this gene, makes the animal to produce more meat. So this is the first, actually the second animal, the first fish considered conventional, a mutation that already exists in nature, and was considered conventional in Argentina and in Brazil now. In Brazil was 3 months ago, in Argentina was December last year. Let me get back. I took out the slide because of the time. But China, Canada, and United States already this same kind of mutations in swine, in ovinus. Ovinus. I'm tired. My English is going down. Sheep, and Nelore race. They already did also. It's not being used commercially, but in terms of research, this same kind of mutation is being done. I don't want to discuss ethics about that here. If you want to discuss ethics, go to China, because there they are using CRISPR for monkeys, for dogs, for everything. They have now a beagle, the dog beagle, with this gene changed. So it's a... In terms of agribusiness, these kind of animals, according to my colleagues, increase around 20% of the production of meat. So Brazil is the first meat exporter in the world. So it would be something that you should look at. We have some colleagues at Embrapa. I'm sure some universities are looking at that. Just for you to have an idea, this is the first transgenic fish liberated to commercial use as food. It's being consumed now in Canada and in US. Took 25 years to liberate commercially, millions of dollars to the disputes that cause, and it's being consumed now. It's safe, people are eating. The salmon grows twice the speed of the regular salmon. And there is many things coming. I'm going to speed up. There is already this transgenic apple being consumed in Canada commercially with RNA, but the plant is transgenic. The plant expresses RNA that turns off some of the polyphenol oxidases. So that browning, when you cut the apple and it start to get dark, that it's reduced. So the commercial value of that product is actually better. They're doing that for strawberries, banana. You have a very tough disease now for bananas all over the world, and people are using CRISPRs to try to delete. The mushrooms, a famous one in citrus. The point is, with this new legislation, investment, in our case in Brazil, doesn't matter if you have a good legislation, if you don't have money to develop those products. And not money only the public sector, the private sector, like in US, like in UK, like in the developed countries, must to understand that they must invest in this kind of technology here. But the point here, there is being a democratization of the use of biotechnology. This data is from Argentina. You can see here, since 2000, this is data from the government of Argentina. Since 2016, when they started with this new legislation, when you look at the GMO crops approvals, most are the 4 big companies. Okay? When you start to look at the new breeding technologies where genome edition is included, you start to see national companies. And I was there 2 weeks ago in two conferences. And it's amazing how the thing is going fast there. The another thing is happening in Argentina. It's a democratization of the crops, because today you have just use of biotechnology for corn, soybean, sugarcane, eucalyptus, and cotton, because they are cash crops. You cannot use this kind of technology for other crops. Or I'm repeating that. This is a blah, blah, blah here. Some of the projects we have at Embrapa. I will not spend much time. Here, just to finalize, this is our fields for GM, so I think for drought tolerance, we have very, very nice results, even for flooding, that's important for here, for the region of Pelotas. But those are transgenics. We don't have $130 million to put in the market. In our case, we are seeking a partner. Grupo Dom Mário is our partner. We're trying to see if we're going to go ahead and try to deregulate. But then we're going to need to Europe, we're going to need to China, we're going to need to the Middle East, follow the air rules, all the experiments they ask for, and the cost is going to be huge. But if it's a good trade, we're going to do that, and we are, whoops. I think it's for me to finish. Anyway, I didn't have more, I just have my information, but those slides will be available. You can have my contact with Dr. Avila and the organizers. And thank you very much, and I'll be glad to answer any question if you have.
Well, thank you, Dr. Alexandre, for your lecture. I would like to invite the speakers to return to the stage. And we have time for questions. Question can be asked in Portuguese or in English. And if you have a specific question, please add the questions for the speaker.
Deliver herbicides or even double-stranded RNA to induce RNAi directly in planta, if it's possible, or you could just use non-encapsulation to deliver it into the soil or in the environment.
Yes, it's possible. I show it just an example using a spray on the plant, but it is also possible to do the delivery in the soil. If you see that article that was published by me and the group of Professor AG was tested, we intend to project a device to release the imazethapyr in soil. It's more complicated to quantify how much herbicide you deliver in the soil, because you have a lot of factors like absorption, degradation, microorganisms. It's not an easy study, but it's possible. For taking into account, for example, the rice culture in flooded land. This kind of device based on swallowable polymers, it's very interesting, because you can use the water to drag the encapsulator herbicide and even to control the release. So it's important too.
There's a publication about bioclay, nanoclay, which was really, the long and short of it is that they used these clay particles in order to stabilize the RNA long enough so that it was able to penetrate deep enough into the plant to mount an immune response. The response then led to increased resistance to herbivory. So there are processes by which, basically, if you can stabilize the RNA long enough, then hopefully then it will create the process that you're looking for, the loss of function or the gain of function, depending on what it is. And some of these, there was quite a huge debate recently at a conference that I was at, was this genetically modified? So again, the type of sprays and that sort of thing, the process can happen, but it's the regulation that comes down to. So the delivery is key, and the results that happen are measurable.
André, is it possible to combine or blend two different herbicides, and after that, when you encapsulate these herbicides, you set different times of release for each herbicide? Let's say a pre-emergent herbicides, you set for the herbicide A to release from the day 0 to day, I don't know, to day 10, and after that the herbicide B will start acting. Is it possible? Have you guys already tested this kind of thing?
That's a very, very good question. In fact, the answer is, I think, is the chicken of the gold eggs, because yes, it's possible. It's possible to combine different kinds of agrochemicals into the nanocapsules. For example, a pesticide, a herbicide, growth factors. So why this mixing will interact into the herbicide? Because this kind of study is not so easy to understand. It's not to the two acts, the first one to encapsulate it, and the second act to release, but to try to understand how the compounds into the nanoparticles will interact. You have a lot of physical and chemical process occurring inside the nanocapsules. It's possible, maybe. You need to study. And this part can be done by the chemistries. For your question is very good, because you have the vision of the agronomy, but there are several other questions that needs to be answered by other expertise.
More question?
Again. So this time I would like to ask a question to Dr. Alexandre. We can see a trend on startups being formed in being started in USA on gene editing on the context of agriculture. So I would like to ask you if you think we will see this trend here in Brazil as well.
The use of genome-edited plants, for sure, it's happening already. In Argentina, like I showed, many startups, companies, they are keeping their young scientists in Argentina and building products and strategies. I saw 3 presentations of startup companies looking at the different solutions for problems in agriculture using genome edition 2 weeks ago. So it's happening in the US, it's happening in Canada, China, like I said, there is many groups working different for medicine, for industry, and for agriculture. And I hope in Brazil we're going to have the same trade. It will happen, coming from overseas, or I hope by technology being developed here. Both actually is going to happen, I hope.
Okay, if we don't have more questions.
Just some a little comment that transgenics is still going to be very important. People sometimes confuse, say, you're saying now genome edition is going to substitute. I don't think so. There is some traits that will be difficult to use by genome edition. The problem is transgenics, GMOs, will be done by only the big companies that have money to do all this deregulation.
Okay. If we don't have more questions, I would like to invite the organization committee to give a little gift for the speakers.